Hello Year 11. This is the last lesson, in the unit B2.3 on DNA and inheritance. Last lesson you had a lesson on cloning insulin, so this just leads on from that, and it's looking at the advantages and disadvantages of genetically modified organisms. So you will need to turn to page 47 and 48 for this work. I'm going to start off first of all though with a little starter activity, a retrieval practice to check your understanding of cloning insulin, because this was done before Christmas. So excuse me whilst I scroll to find the right page. So up on the board, current slide, you can see some cloning insulin questions. You could write these on the back cover of your B2.3 booklets. So pause the video, read the 7 questions and see if you can answer them. So hopefully you've answered those questions. So we've got question 1, what was cloned in the experiment? We managed to clone insulin. What is cut out of the human cell? It is the insulin gene. What does the cutting? It is an enzyme. Why are bacteria used? Now there's 2 reasons, It says, as a hint, think about their size and reproductive rate. So they are very small, so they take up very little space. So in terms of culturing these bacteria on a large scale, you could have huge vats of the things, to create quite a lot of product. Their reproductive rate, they reproduce by asexual reproduction, which is creating exact clones. And each of these bacteria, probably they divide once every 20 minutes, most bacteria, if they're in the right conditions. So their reproductive rate is very, very fast. So you wouldn't, for example, think of putting a gene into a human, because it would take 9 months before the human was created, because that's the gestation period. So the benefits of this process is that it makes it very, very fast, and it makes it very efficient, because we're creating a lot of the product in a very limited space. Disadvantages of producing genetically engineered insulin. Some people do have concerns about the safety. Every batch of insulin is always tested, because that insulin could be contaminated with bacteria that could cause illness. And obviously that insulin would not go out and be distributed to the public, to patients that require it, unless it had been tested and was clear and free from that. A similar experiment was also performed to produce a growth hormone. This growth hormone is used to treat a condition called pituitary dwarfism. So your pituitary gland releases growth hormone to help you grow, particularly as you go through your teenage years and through puberty. Before the hormone was created using genetic engineering, the condition used to treat people with pituitary dwarfism was to extract growth hormone from the corpses of unaffected people by removing their pituitary gland and grinding it up, and using that to inject. However, some children developed a disease of the nervous system called Creutzfeldt-Jakob's disease, CJD, so its use was continued. So think about how they would use the same process in cloning insulin, to be able to create the growth hormone. So if I go back to the diagram on cloning insulin, so probably this one here. Instead of taking out the required gene as the insulin gene, this would be the gene for the growth hormone. So that would be removed from a human who wasn't suffering from pituitary dwarfism. You would take your bacteria, and the bacteria we normally use are E. coli. Inside the bacteria is a ring of DNA called a plasmid. That plasmid is cut open using enzymes. The gene for the growth hormone is inserted in, and it's glued in using the same kind of enzyme. The recombinant DNA is popped back into an E. coli that hasn't got the plasmid, and then that E. coli bacterium is allowed to start to divide and reproduce. So it's put into some growth media to mass produce, okay? Instead of insulin, this would be mass producing the growth hormone. So see if you could write a flowchart to describe how that growth hormone could be created on a mass scale. So to today's lesson then. As I said, page 47, 48, there's a table to create first of all, showing the advantages and disadvantages of genetically modified organisms. And then this leads into you being able to produce a balanced argument text. So taking some of those statements and actually putting them into extended prose answers. So in theory, this process could be used to mass produce any protein. In 1993, the first protein was genetically engineered, okay, using this method. And it was actually a blood clotting protein. People who suffer from haemophilia find that they don't make this blood clotting protein. And so for you or I, if we cut ourselves or if we knocked ourselves against the side of a desk, we would get a bruise or a cut, but our blood clotting protein would thicken the blood and would stop you carrying on bleeding. With haemophiliacs, this is not the case. They don't make this blood clotting protein, and so as a result, they can end up bleeding to death. So the way the haemophiliacs were treated was they took this blood clotting protein gene, they introduced it using genetic engineering, very similar to what we've just talked about with insulin and the growth hormone, and they introduced it, instead of into bacteria, they put it into sheep cells, and made the sheep cells start making the blood clotting protein. And then the haemophiliacs were treated with this injection of this protein to help their blood to clot. So in theory, this process could be used to mass produce any protein that we want. We also have genetically modified crops, and the very first commercially genetically modified crop, GM crop, was modified so that it was resistant to insects and pests. What we mean by this is that insects didn't eat it. So it was the potato, and the potato was modified using genetic engineering to have its own built-in insecticide. So they took a gene from another organism that was a known natural insecticide. It was known to kill insects if they ate it, and they introduced it into the potato. The potato itself was completely harmless to humans when they ate it, but if insects ate it, they died. So it saved the crop, okay, protected the crop, so you had a greater yield, a larger yield of crop. So how are crops genetically modified? Well, we've actually been doing genetic modification for centuries, okay? Traditional breeding methods, we choose to cross different species, or same species, but different types of the same species together to give us the desired features. So let's say if we've got one strawberry plant that produces lots of strawberries, and you've got another strawberry plant that produces big strawberries. If we breed them together, you'd like to think that the offspring would produce lots of big strawberries. So this is what we call traditional breeding methods. But there are some other ways that we can genetically modify crops. There's a method called mutagenesis, and we're going to come across mutation in the next booklet. What mutagenesis means is we expose the seeds to either chemicals, mutagenic chemicals, okay, or to radiation. And the radiation and the chemicals cause the genes to change, to be altered, just as if we were exposed to radiation and certain mutagens, our genes would change. These changes are random and they're usually unpredictable. There's no safety testing requirements for carrying out mutagenesis. So this is one of the reasons why people are concerned about eating genetically modified crops. The second method is called RNA interference. And in this method, you'll see it in some of the examples we're going to look at later, you can switch off selected genes with this RNA. So, for example, if there's a gene that causes a particular change in an organism, okay, that gene can be switched off or silenced. There is safety testing required for this. So you'll see that particularly in salmon, okay, why they are using RNA interference, okay, and switching off the genes. The last way genes are modified is the method that we just talked about with cloning insulin and creating the blood clotting protein for haemophiliacs. And this is by inserting selected genes using recombinant DNA methods. So you would take the gene from one organism and you would put it into another organism's, DNA. So this is what we mean by recombinant DNA. It's recombined the DNA. The desired gene is inserted only at known locations, and this definitely needs safety testing required. So these are probably the two methods that are used the most in the crops that are genetically modified these days, okay? But just to give you an understanding of the number of genes that could be affected, well, anywhere between 10,000 to 300,000 genes are affected by traditional breeding methods, just to give you a balance of opinion. So this is how we breed crops and how we've bred crops for centuries. Mutagenesis, there's no way to assess the number of genes that have been affected because of the randomness of the changes. In RNA interference switching off genes, it's normally only 1 or 2 genes that are actually affected that are switched off. And in transgenics, it's normally only between 1 to 4 genes that are inserted at particular locations into DNA, normally into a plasmid, into a bacteria. So I'm going to go through some case studies just to give you some examples of the way genetically modified organisms have been created and for what purpose, the benefits, but also some of the disadvantages. So this first one, is particularly significant because some countries, have problems growing enough food for their population, but also they may have problems with pests. In the past, we have sprayed crops with pesticide or insecticide to kill the pests, but we know that these chemicals are not good for us. They enter the food chain, they might enter the human food chain, and they cause damage further down the line in food chains. So if we could take the gene for a natural insecticide out of a bacteria, and then we could introduce it into a very young wheat plant when it's still just like an embryo, then basically that plant will grow up to naturally release an insecticide that is harmless to humans, but when insects start nibbling on this wheat, they will die. So it saves the crop, it ensures that the crop is going to be a large enough crop, It's not going to mean the farmer's going to lose any money. So this is probably the biggest, most significant type of genetic engineering, that is an advantage. Case study 2, this is quite an unusual one. The person who came up with this idea is a very, very creative idea. Strawberries generally only grow in the summer, so we have a very limited season in which to grow them in the UK. We can grow them in greenhouses under artificial conditions. However, we cannot freeze them particularly very well. So even if we mass produce lots of strawberries, the idea is that if we could freeze them, and then when they're defrosted, they look exactly the same, then we don't need to grow them at any other time of the year. So strawberries always go mushy when they're frozen due to the cell walls breaking down. So they're only really useful for making jam or strawberry sauce. There are some fish in the Arctic that produce a natural antifreeze in their blood to stop themselves freezing. If we could take this gene for the antifreeze from the fish and introduce it into the strawberries, then the strawberries will not go mushy after defrosting if they're frozen. The strawberries will not taste of fish, and as a result, it means that we can grow these strawberries in the summer, we can freeze them, and then they can be defrosted at any point in the year rather than having to try and grow them outside of their seasonal growth period and using a lot of energy in terms of lighting and heating to grow them when they shouldn't do. Case study 3. We're looking at a combination of genes in a daffodil produce vitamin A. However, I do not advise that you start eating daffodils because they are poisonous and toxic. They won't kill you, but they'll certainly give you a very upset stomach. We know that in some developing nations, the only food that people may be accessing is rice. And so because rice does not usually produce vitamin A, lots of the population may be deficient in this particular vitamin. For children, this is a big problem because deficiency in vitamin A can lead to high child mortality. In other words, the death rate increases. So if we could take the genes for vitamin A from the daffodil and introduce them into rice grains, then we can make these rice grains start to produce vitamin A. And as a result, you would get your vitamin A from your rice that you're eating. Case study 4. Crops of the legume family, things like peas and beans, possess natural colonies of a nitrogen-fixing bacteria in the nodules of their roots. So people who are studying Triple Award may remember studying this back in year 10. These bacteria can take nitrogen straight from the air and form it into nitrates to help plants to grow. So if genetic engineers could introduce this ability to take nitrogen straight from the air into other crops that can't do this, the economic implications would be enormous. This would mean that we wouldn't need to use fertilizer to help increase the growth of our plants. So the homegrown bacterial fertilizer, unlike the artificial fertilizers, will not cause pollution or enter the food chain and cause problems in that way. So these have all been so far advantages. This next one is also an advantage, but it does have a disadvantage in it. Scorpions have a gene in one of their chromosomes that makes a protein that's poisonous to insects. So it's a natural insecticide, if you like, from the scorpion. This gene can be removed from a scorpion and inserted into the nucleus of a fertilized egg cell from a cauliflower. Each time the egg cell divides, it makes a copy of this scorpion gene as well as copying its own genes. As a result, the cauliflowers produced will have the poison in their cell, which acts as an insecticide and will kill the caterpillars that eat the cauliflowers. The scorpion gene is harmless to humans, It's not that we're eating scorpion poison. However, if we kill all of the caterpillars that are eating the cauliflowers, then we'll have a reduction in the local butterfly population, and this will have an impact on the rest of the food web. So, disadvantage is that by taking out the caterpillars, by removing them from the food chain, it will impact, the rest of the food chain, particularly the butterfly population, and butterflies are responsible for helping with pollination of plants in a local area. So, but there is an advantage in that a natural insecticide has been incorporated into our cauliflowers, so our farmers will be able to have more produce and make more money. Case study 6. This is a really good example, of how we can go about treating cystic fibrosis. Now, in the next unit, B2.4, we'll cover an entire lesson on the inheritance of cystic fibrosis and the treatment. Researchers have already identified the faulty gene. It's actually a series of genes, for cystic fibrosis. It's not just one. This is a disease that affects humans' respiratory and digestive systems, You cannot catch this disease. It is an inherited disorder. And basically, scientists have been able to isolate the non-faulty version of the gene in a non-sufferer. I don't like to call people who are non-sufferers normal because none of us are normal. So a non-sufferer is somebody who hasn't got cystic fibrosis. And they can insert this non-faulty version into a virus. Now, you might be thinking, well, that sounds a little bit on the dangerous side. This particular virus is what we call a non-virulent cold virus. In other words, it doesn't cause us to develop the symptoms of a cold. The virus can then pass into humans by using a nasal spray in the hope that the lungs will take up the gene and stop the excessive mucus production that's associated with this disease. So with CF, sufferers produce so much mucus that it stops oxygen getting into the lungs very easily. It stops absorption of food into the gut. And it's a debilitating disease, and it can get progressively worse, damaging the lungs and the digestive system at each episode. So we call this gene therapy. So we could use this gene therapy for altering any genes that are affected by genetic disorders. Okay, so this is another advantage. This is one where I talked about the switching off of genes. This is case study 7, and it's about wild salmon. Now, wild salmon are basically very, very expensive. So people have been farming salmon in huge, big crates in the water, and they find that the wild salmon have a tendency to jump out of the crates and try and swim back to their original breeding ground and escape. So fish farmers have designed farmed salmon to suppress the genes that control the homing behaviour, so they don't know where to go back. However, this is where we have a disadvantage. If the farmed salmon escaped, they could introduce the mutated gene into the natural population. This could destroy the natural homing instinct of salmon and drastically reduce their numbers, leading to their extinction. So sometimes by introducing this suppression of genes, okay, if these genetically modified organisms do escape into the environment and breed with the native ones, okay, we can lose that ability for salmon, for example, to naturally home, okay. It's like they've got their own little sat-nav built in, okay, to find their breeding ground to go back to. So that's another disadvantage. So some other reasons why people are against genetically modified organisms. So this is opposition. The main concern that people have is that if we're genetically modifying our food, that the animals, including us, that eat these crops that are genetically modified may transfer antibiotic resistance to the bacteria in their gut. So this has been investigated, looking particularly at cattle, how many of them have antibiotic resistance. You may have heard of this disease called MRSA. So it's feared that this could add to the spread of antibiotic resistance in the world, in turn leading to more superbugs like MRSA. Again, in the next unit, we're going to learn more about how MRSA has developed. But it's natural selection of bacteria that are resistant to the antibiotics. In other words, they don't get killed by the antibiotics. So therefore, they survive and they can divide and multiply and take over a body very, very quickly. And in particular, in people who are elderly, who are young, who are weak or vulnerable because they've got other infections, MRSA can be a potential killer. Mutated genetically altered species could also escape, as we saw with the wild salmon, and compete with native species, causing them to become endangered and potentially leading to their extinction. So if we've got a crop, a native wheat crop, okay, and a genetically modified insecticide crop has been grown nearby, if this crop is more successful, it could outcompete our native crop. So we could lose species. Genetically modified crops, though, are often not fertile, and this means that they are less likely to reproduce with the native species. This in itself, by making them not fertile, which is an advantage, a good thing, causes a disadvantage, because if they're developing countries that want to buy some of this wheat that's got this genetically modified insecticide gene in it, they would have to buy new seeds each year. They can't just use the seeds from the crop because they're infertile. Another disadvantage, there are concerns over insects becoming pesticide resistant if they eat a constant diet of pesticide-forming plants. People have concerns over the long-term effects on the health of humans who eat these GM foods. And I've got to say, if you Google online, you will find that there's a whole variety, particularly snacks, things like I'm not going to name particular brands, but crisps and things like that, that do include ingredients from GM crops. And these foods have been in our supermarkets for decades now, okay? So we are still looking at the long-term effects on the health of humans. Finally, public concerns that if we can manipulate the genes of plants and animals, then we can manipulate the genes of our future children. So the idea is that people who had enough money could create what we call a designer baby, okay? And what we mean by this is that they could have the genes of the baby altered to make the baby super intelligent, super attractive, okay? Tall, blue eyes, blonde hair, whatever features you felt was the desirable features, okay, to create this designer baby. But this would create a two-tier system where the people with money would be able to create the perfect children, okay, and the people without money would not be able to create them, so it would be a natural selection causing the offspring to be slightly different. So I need you to make sure that you're filling in the table. You can either have been pausing that and writing them down as we went through them, or you could be pausing it and writing down what you think, okay, are the advantages and disadvantages. So I've put a couple on here just so that you can see. So plant can be modified and tailored to grow in any region of the world. So it could be drought resistant. They could be used to produce biofuels to help ease the fossil fuel crisis. Disadvantages, okay, they would need to keep buying the seeds every year, the developing countries, and antibiotic resistance, okay. Once you have filled in your table, you can use this then to produce your balanced argument text. So I know in English, okay, you have a variety of different sentence starters that you use. So it might be on the one hand, on the other hand, the advantages, the disadvantages, the pros, the cons. So you know you can use those sentence starters to produce your balanced argument text, okay? Make sure that you're not just writing about advantages or disadvantages. Make sure that you've got a balanced argument. So that is the last lesson in the B2.3 booklet. Thank you.